Vamos dar início então, pessoal. Bom dia. Nós, então, naquela ideia de aproveitarmos os visitantes externos para o uso do nosso Grand Round, nós vamos ter hoje outra sessão especial, que é muito bom para nós, sempre receber alguém de fora, que realmente nos traz as novidades que estão ocorrendo aí por fora. E hoje, então, é o serviço de genética médica, que nos traz um convidado do exterior, e ele será apresentado pela professora Lavínia.
Bom, eu queria agradecer, então, em primeiro lugar, a presidência do Hospital de Clínicas de ter proporcionado essa oportunidade e apresentar não só o Jeffrey, mas a importância do tema também. Então, eu vou rapidamente falar para vocês. Vocês devem ter visto no anúncio que nós fizemos sobre esse Grand Round, que nós estamos comemorando 200 anos do nascimento de Darwin. E quando a gente fala em genética de populações, ou quando a gente fala em genética, tem aquela história que é verdadeira, a genética não faz sentido senão à luz da evolução. Tá? Então, hoje, o professor Jeffrey Long, que é da Universidade de Michigan e que tem uma trajetória bastante longa de colaboração com o nosso grupo de genética de populações aqui em Porto Alegre e agora conosco no Hospital de Clínicas, na parte de genética médica populacional também. Ele é, nesse momento, professor da Universidade de Michigan e está se mudando para o Novo México agora, onde ele vai começar uma nova vida lá, mas continuando com seus temas principais de genética de populações. E ele traz, então, para nós um tema que é muito importante, que está sendo muito discutido, que é o conceito de raça. O que isso significa em termos de genética, se existe ou não, mas mais importante para nós, não é uma questão conceitual, mas como é que isso se aplica na prática clínica, especialmente em doenças comuns. Nós não vamos falar não vai falar quase de doenças genéticas, mas sim na aplicação disso para o clínico que está trabalhando com doenças prevalentes. Jeffrey, thank you very much. Please, you can go there and start your lecture.
Thank you very much. It's a pleasure to be invited here to speak. This podium is very impressive. I feel like I should maybe give a sermon instead. But it's always a pleasure to come to Porto Alegre. This is my third visit, and hopefully this seminar will have something to offer to the audience. Just starting point, I think that it's important to realize that scientists in the United States and other countries, certainly in Brazil, have argued about race for a long time and really over 100 years. And despite all of this concern that people have expressed, we have many questions still, and they're fundamental questions. What is a race? How is membership in a race determined? How many races are there? How do races evolve? And then I think the most important question is, does membership in a race convey any useful information about a person? So when we assign people to races or assign ourselves to the membership in a race, we usually do that because we want to convey some sort of information, either positive or negative, or some statement about our ethnicity or the like. But it's not really clear that race is a very good proxy for the things that we intend it to be. Well, after this 100 years or so, things have changed a little bit in the last decade because of human genome sequencing and the Human Genome Project. And there's been some feeling that all of this information about DNA sequences will answer some of these questions. And that's really the basic topic that I would like to look at today. So, first of all, this is pretty fundamental information here, but just for a quick overview, I'd just like to say that the human genome sequence is composed of the DNA molecule and there are actually over 3 billion nucleotides in it. So anytime that we talk about genetic variation, we're talking about not only a sample of people that have been in our studies, but also a sample of the genome that we're looking at. Most of that genetic variation is non-coding, and a lot of it is just repeat sequences. Only about 2% of the genome encodes genes and sequences with functional products. One of the things that's most amazing to me is that 10 years after sequencing the entire human genome, we still don't know how many genes there are in the human genome. I know when the genome sequence was completed, my relatives asked me what I was going to do since we knew everything at that point, and yet it was just a starting point even for the most fundamental questions. It's hard to even say what a gene is. Some of them are very small. 75 nucleotides would be the smallest. Some of them are very large. 2.5 million would be the largest. And most genes have alternative forms called alleles. But most of all, when we talk about variation, I think the sort of one take-home number that we can use is that if we compare two copies of the genome, there's a difference in the encoded information at about 1 in every 1,000 or a little bit more, 1 in 1,200 nucleotides. So by that, when we talk about comparing genomes, sometimes we think about comparing people with each other, but as a geneticist, I compare genomes. So I think about it, if I were to, I inherited 2 genomes, one from my mother and one from my father, and with the exception of the sex chromosomes, I've got a copy of the same thing from each parent. But if I looked at it, there would be millions of differences between what I inherited from my mother and what I inherited from my father. And that's genomic variation. And then the next question we have is when we look at that sort of variation, I've described how it's important to me, but how does it play out in populations as well? Just to take a quick look at a variation, this was done by Aravinda Chakravarty, who was my postdoctoral advisor a few years ago. But he DNA sequenced about 12,000 nucleotides from genes that are related to blood pressure and risk for heart disease. And out of that 12,000, and this was in 75 people or 150 copies of the genome, there were a total of 58 positions that where there was variation at all. So it doesn't seem, and that's consistent with the earlier numbers that I gave. So it doesn't seem like there's a lot of variation, but we can put that into a little bit of perspective by comparing it to what would happen if we compared a human genome to a chimpanzee genome. And as a geneticist and as an evolutionary biologist, to me, that's almost the same thing. But what's interesting here is that of these almost 13,000 sites, about 12,850 of them are exactly the same between humans and chimpanzees. So that this small amount of variation here is about equal to the number of places in the genome where sometimes at 53 of these sites, the humans had one allele, every human was alike, and chimpanzees had another. So when we talk about variation in the genome, we're talking about essentially rare events if we're talking amongst ourselves or even with comparing different species. Now, to more closely relate to questions about race and the pattern of human variation, I'd like to talk about 2 data sets. And one of them was collected at the University of Chicago by Wensheng Li in his laboratory. And what he looked at were 50 very small loci, each about 500 base pairs apiece, of non-coding DNA. And he looked at a sample of 30 people, 10 Africans, 10 Europeans, and 10 Asians. And this was the first unbiased representation of nucleotide sequences in people from different populations, in humans. And this was published about 7 years ago, in 2002. And the first thing that was of interest is if he looked at the nucleotide diversity which would be essentially the chance that if you looked at a particular nucleotide position, a person would be heterozygous. If you looked in Africans, there was about twice the nucleotide diversity that there were in either Asians or Europeans. And Asians and Europeans were quite close to each other. So that was a pretty startling finding, to have so much more variation in Africans than in Asians or Europeans. But if he compared one genome from Africa with one with Asia, well, there was certainly more variation than if he compared two from Asia or two from Europe. But what was interesting here, and he got the same pattern comparing Africans to European copies of the genome, was that there was actually more variation when he compared two copies of the genome from Africa than if he compared one from Africa with one from Asia, or one from Africa and one with Europe. That was a pretty startling finding, and I don't think I would have predicted that. I don't think anybody else would have predicted that either. If he compared two copies of the genome, one from Asia and one from Europe, there was a little more variation than if they were both from Asia or both from Europe. But in fact, that increase in variation is not even statistically significant. So one of the most fundamental parts of understanding human genetic variation at the DNA sequence level is this enormous variability in Africans, and if you compare it across Africans, and the reduced variability outside of Africa. Now, there's only been a few studies at this level of DNA sequence in Native Americans, and Nelson Fagundes, who's sitting here, conducted one of them. I apologize for not in putting his data into this slide as well, but I thought it was easier if I just stuck with the published study, the first published study. Now, another way that I would like to look at of thinking about the same data is on this slide, where I've redrawn the information and used a Venn diagram to describe it. And basically with this Venn diagram here, the idea is that there's a lot of variation in Africa, and only a subset of that variation is in either Europe or Asia. And the subsets in Europe and Asia are highly overlapping with each other. And this gives us the fact that if you draw two from Africa, there would be more diversity than if you drew one from Africa and one from Asia. But this idea now, when we want to think about how human genetic diversity and DNA sequence diversity is distributed throughout the world, rather than thinking about it, and my argument will be throughout this talk, rather than thinking about it as races, we're better off thinking about it in terms of these subsets. So that study in 2002 I thought was very provocative. And of course, it was based on a small sample of the genome, even though it was impressive in its day, and a small sample of people from throughout the world. And I wanted to know, do other DNA data sets show the same pattern of variation? And the best way for me to address that question was to collect data in my own lab on this. And so we looked at a few more loci. We looked at 63 loci with a total of 38,500 base pairs. Some of the loci were the same as in the Wencheng Li study, and some were different. But we picked 3 African populations here, and we looked at 4 people representing each population. And we looked at 5 Eurasian populations, once again 4 people from this, so that we had a very balanced sampling design here. These are how the populations are distributed throughout the world. I tried to get some spread. All of the black dots are populations I'm going to talk about today, but we've also, since I prepared this talk, completed sequencing in 2 more. And I recently got some funding from our National Science Foundation to actually now look at another 15 populations throughout Eurasia as well. So this is an expanding project. But how did our results turn out? Well, first of all, out of those 38,000 base pairs, there were only 204 sites where there were different alleles. So most of the 38,000 of the 38,500 base pairs, about 38,300, everybody was the same throughout all of those samples. Of the variable sites, the vast majority of them, we saw only one copy of the rare allele. And then for another bulk of the sites, we saw either 2 copies of the rare allele or 3 copies of the rare allele. In only 86 sites out of 38,000 did we see 2 alleles at relatively common frequencies. Now, what was interesting here is when we looked at the distribution of the rare alleles and the common alleles, what we found was, first of all, if we looked at a common allele, which meant that it had a frequency of over 15% in our total sample worldwide, it was almost always found in both the African samples and the Eurasian samples. So any common allele was found on both continental regions, Africa and Eurasia. There was one common allele that was only in Eurasians, one common allele that was only in Africans, but as a general rule, common variation is shared by people all over the world. If we looked at rare alleles, we found the effect that Wencheng Li found. There were many more rare alleles in Africa than there were in Eurasia. It was the moderate frequency alleles that were of most interest to us. So there were about 20 that were found only in Africans, and fewer than 10 that were found only in Europeans, and about 18, I think, that were found in both Africans and Europeans. But the overall effect of this is that if we look at an allele with a moderate frequency and it's in Eurasians, either this orange column or this blue column, the chances are that it's also going to be found in Africans because this blue column is higher. If we look at a common allele or a moderately common allele in just Africans, which would include this green column and this blue column, the chances are it will only be seen in Africa. So there's a vast store of genetic variation in Africa. It's the rare and the moderate frequency alleles that Africans have, that non-Africans don't have. Common variation is shared throughout the world. Well, what accounts for this pattern? And I'd like to look at a couple of different things. Well, first of all, before looking what accounts for the pattern, you can link that data in the use that data to link the populations together into a tree-like form. So down here we have the 3 African populations, and then there's a large branch jumping up and linking together all of the Eurasian populations. And what we see now is if we compare, there's not too much diversity in here, but if we compare to Africans, for example, there's much more diversity, but even according to this graph, there's more diversity amongst the Africans in themselves. The goodness of fit of this tree is illustrated in this plot, and I won't go through the details, but it's very good. The R-square is high. It's over 0.94. Okay, so now, what accounts for this pattern? And what I'd like to point out is there are 2 things. The first of all is we share all of the common variation because that common variation is old. And the second point is the pattern was created by founder effects and population bottlenecks that occurred as humans migrated out of Africa. And so this graph here shows us something about the age of common alleles. So if I looked at an allele that had a frequency of 35% in a sample of Han Chinese, I can ask the question, when did the mutation arise that created that allele? This scale here is a little bit difficult to read because it's on a logarithmic scale, but these arrows indicate particularly important points. This point right here, it corresponds to about 200,000 years ago. And what this means is any common allele that I would find in Han Chinese or other Eurasians probably arose a long time ago, even before humans were a species ourselves, but certainly in Africa. And then those alleles were carried through the migrations to the rest of the world. This little blip right here actually corresponds to the time period where people migrated out of Africa. So this scheme here really tells us about the distribution of genetic variation, that 200,000 years ago the species arose in Africa, and that there was a lot of variation in the species, but about 100,000 years ago a small group of Africans left. They had only a portion of that variation, not all of that variation, and they moved into this area here. Eventually, another small group migrated up into Europe, another into Asia, finally into the Americas and into the Pacific Islands as well. So that if we want to understand the distribution of genetic variation and have some framework for thinking about that distribution of variation, it's actually the history of our species and how it migrated from a small species that was exclusively in Africa to being a large populous species throughout the world, with just about the most distribution of any mammalian species. So now we can ask a question of, we've looked at these patterns. I think they're very clear. We have two unbiased representations of the genome. We would like to have larger sample sizes. We're working on those right now, but we can say, how well do race concepts fit this pattern of variation? And that's actually a difficult question to ask and answer, and I was surprised when I started doing this research because I couldn't find definitions of race. And I would go to different textbooks and they would tell me things like, if you think race is this, you're wrong. If you think it's that, it's wrong. But I could never get anybody to say what it was. Eventually I found some. One definition I call the implicit definition, and the idea here is that people like James Crow and Ernst Mayr have used it recently, where they say basically, we all know what it is, so we don't have to define it. And I don't think that that's very productive. There are purely social categories or social definitions of race, and these are both from the United States and may not apply to Brazil very well. But in the United States, we had something called the one-drop rule, and that any traceable ancestry to a subordinate group put you in that subordinate group. And that was a government policy that was created in the 1850s so that they could decide who was a slave and who wasn't a slave, and settle property issues. Currently, our government has an Office of Management and Budget, and whenever you apply for a government grant or you do any sort of government census work, they have categories of race that we're required to record. They're rather broad categories and maybe not even scientific categories, so they're categories that would be like non-Hispanic white or black or something like that. Finally, biologists such as Dobzhansky have talked about populations and races, and basically populations are races are populations whose members are more genetically similar to each other than they are to members of other populations. And so this is an interesting definition here, and it's one that we can actually work with and say, does this definition then give us back the races that we're used to seeing, like Africans and Europeans and Asians? So this is typically the population concept, that you have overlapping groups, but this population concept doesn't really correspond to what we've seen both in my study and in Wenxiong Li's study. And I'd also just like to point out that when we use the traditional categories, here we have the African population sort of in Africa, the Europeans in Europe, and the Asians in Asia, versus what we've seen here, what we find is that traditional category sort of hides the variation, and also that it doesn't fit the data as well as this expanded category here. So these are a couple of statistical analyses, and I'll just point out that if we use the traditional race groupings, which I've called here a two-level island model, and I compare it to the expanded tree, basically there's a significant, we can reject the hypothesis that both of them account for the data equally well. And we can use a different, this is a F test, we can use a different criterion based on information theory as well. Nevertheless, I should say that either one of those versions, whenever you make a tree, whether it's a simple tree like racial groupings or an expanded tree, they don't account for the data perfectly, and you can reject any model that you use. So I always go back to this quotation from the statistician George Box, who said that essentially all models are wrong, but some are useful. And my argument here is that when we go from traditional races to the more expanded trees, we're moving to something that's more useful to us. But I'd just like to point out also that the notion that we can divide people into broad groups like Africans and Europeans and Asians doesn't fit this data very well, because if we look at the smallest group that includes all Africans and all of the variation in Africa, it includes everybody in the world. So if we use the biological definition of race, Africans are not a race. And if we were to define a race, then by shared ancestry, it would be along this branch here, but it wouldn't break out into a traditional race like Europeans or Asians. It would break out into Eurasians as a single group. And then if we wanted to look at the traditional races, we would end up with sub-races. So that using traditional race classifications to describe our variation doesn't have a lot of utility, because one racial group that we've identified throughout history, or Africans, would not be a race genetically whatsoever. Another group, Europeans, that we've identified as a race throughout history, would be considered a sub-race. So I think it's easier just to think in terms of the patterns of migration out of Africa, and these trees. They're still visual, they still tell you a lot about the distribution of variation, but the sort of taxonomies aren't very useful. Okay, well, one of the questions that we have is, with this pattern of variation, can genetic markers identify populations? And the answer to that question is yes. So my colleague at the University of Michigan, Noah Rosenberg, did a study where he did a cluster analysis, and he looked at 52 populations from throughout the world, and he tried to put them into 5 groups here that he labeled with colors that people have never come in. But basically what he found was each continental region identified with one of these groups, with the exception of South Central Asia that was distributed throughout. So it is possible to place people in clusters. But when we look at these clusters, we have to ask questions about what they mean. This is from a study by another colleague, June Lee. And what we find is it creates people as mixtures of what they call major groups. But you can see, for example, in this brown group here, nobody is a pure member of that group. So it's sort of a fictitious group. And some of these mixtures don't make sense. Like in this column here, it would put Russians, and in this case the Russian sample is from Moscow, as being a mixture of other Europeans and Native Americans, which are quite a different ways away. And the idea that the people in Moscow have had some Native American mixture, I think, is a little bit far-fetched. My colleague Kenneth Weiss and I have been trying to look for alternatives to that. This is maybe a little bit complicated diagram, but here we've just compared DNA profiles using the forensic markers like you would use for either linkage analysis or human identification. And we have a square here that are comparing 100 people with everybody else in the sample. So all pairs of the 100 people, 10,000 squares. But what we find is that there's very clear patterns of the homozygosity in the sample. And you can see groups, but you can also see how the groups are related to each other. So we found this sort of the previous one here. It doesn't, even if these groups are valid groups, it doesn't show how the groups are related to each other. Whereas here we can see there's a band that corresponds to Africa, and then you see Eurasians coming out, and Asians, and Native Americans. One of the other things that we found in this approach is there are a few dots that are off the diagonal that are, have these dark red and blue colors, and those are actually relatives that appeared mistakenly in these samples that were supposed to be random. So we think that there are other ways to go about this. The other question is, if genetic, and this gets, I think, to something that's most important, if genetic markers, if I can look at, say, 6,000, 600,000 genetic markers and assign people to populations, can I use somebody's population measurement to say what their genotype would be? So on the one hand, we can go from genes to population, but what we really want to know is, can we go from population to gene? In other words, if I know that somebody's Italian, do I have to genotype them or something? And this is a study from a study that I did a number of years ago, where we were looking at an allele at the aldehyde dehydrogenase 2 locus. It's the one that we were interested in was this red allele, and it only appears in Asian samples, so from Mongolians to Cambodians, Chinese, Taiwanese, Koreans. And what's interesting to me here is if somebody has this allele in their genotype, you know with certainty they have Asian ancestry. But if they have Asian ancestry, the chances are they will not have this allele in their genotype. So I think that there's a mistake that people are making right now, which is they're trying to say that because we can use genetic markers to identify populations, we'll be able to use the population to identify genetic markers. Or to put it in the context that might arise in medicine, if you told me that there was a person standing behind this curtain here and that they carried the sickle cell gene, I would bet a lot of money that they were of African descent. I might lose that bet because they might be from South Asia, but most likely it would be of African descent. On the other hand, if somebody was standing behind this curtain and you told me that they were of African descent, I would not bet that they carried the sickle cell gene, because most people of African descent do not carry the sickle cell gene. And I think that has a lot of relevance to medicine, because in medicine we collect information about people's ethnicity, but how can we use that information? Does it tell us something about the genotypes that they'll have or the disease risks that they'll be running? I'd also just like to point out, this is from a study that Carrie Pfaff and Jill Barnholtz, who used to work in my lab, conducted. It was a purely mathematical study, but we were looking at ancestry informative markers. And in ancestry informative markers, I'll just point out that if we looked at the population, a mixed population that was mostly European with some African ancestry, these light blue markers are very informative, and these green markers are not very informative. But if we looked at a different population that was mostly African with some European ancestry, what we find is the green markers then become informative and the light blue ones are uninformative. So we talk in the literature, you'll see a lot of concern with what are called ancestry informative markers, or AIMs, and even those have to be used carefully, because what's an ancestry informative marker in one population is not an ancestry informative marker in other populations. And we've basically been unable to find markers that work universally as ancestry informative. The last topic that I would like to touch on is the question of, does genetic ancestry predict our disease risk? And I'm just going to draw on some information that my colleagues and I from the National Institutes of Health looked at for diabetes in Native Americans. And in this study, we looked at European ancestry in people of Native American origin. And so this was conducted on a North American Indian reservation called the Gila River Indian Reservation, which is in Arizona. And what we found here that was quite interesting was that that the higher, as your fraction of, if you were mixed, the higher your fraction of European ancestry was, the lowest the odds ratio or your risk for diabetes was. So it was a marker, if you will, of protection from diabetes. And that was of some interest to us. This was based on purely diabetes, which is very prevalent in the population, but we looked at other variables related to diabetes, such as body mass index, and we found the same trend there. But when we did a further analysis of it, we got some interesting and unexpected results. So first of all, since we were looking at diabetes, we had age and sex in all of our models. And if we estimated a person's ancestry from their genetic markers, what we found was that the odds ratio was below 1, indicating that European ancestry protected from diabetes, and it was statistically significant in an important way. But if we estimated their ancestry just from what they told us about their relatives, so we asked most people about their parents, their grandparents, and their great-grandparents, if we just used what they told us, we got a better predictor of their diabetes risk. And we were going to stop there, but we hypothesized that what you know about your ancestry, if it's all about genetics, then all of the information from the genetic markers should go away if you put both into the analysis. But when we used both, what we found is that both of them were statistically significant. So that there was a significant effect of measuring somebody's ancestry from their genes, as well as just from asking them about their ancestry. And we've interpreted that to mean that there's a correlation between ancestry and life ways. So even among these Native Americans who all live in the same location, on the same reservation, go to the same schools, go to the same hospitals, have very similar life ways, there seems to be some correlation between their ancestry and their lifestyle. So with that, I'd just like to pull together a few threads from the talk in general. The first thing I want to point out is that rather than talking about whether races exist or don't exist or whatever, I think that the real issue to us as biologists and as clinicians is to understand how human genetic variation is distributed. And there is a lot of human genetic variation, and it's distributed in these what I call nested subsets. So you can think about it like the Venn diagrams where you have circles inside of circles, or you can think about it like the Russian dolls where one doll fits inside of another doll inside of another doll. When we want to explain that pattern, it's best explained by the migration of people out of Africa into filling new continents, such as the Americas, and subsequent mixtures as well. It's not explained well by our traditional race categories. Ironically, our genes predict ancestry better than our ancestry predicts our genes. So in other words, we can look at particular markers like the ALDH2 marker and take that one marker, and if we hit the right allele, we can say with certainty somebody's Asian, but if somebody's Asian, we can't say with any certainty what alleles they'll carry. Health and disease risks are predicted by ancestry. I just gave you the one example of the diabetes and Native American ancestry. There are many others, but I didn't want to go through all of those. But even in that case, our ancestry, or what we would say about our ancestry, often confounds genes. And here I've used environments like geneticists use environments, but really I mean lifestyles and habits. So finally, I would just like to thank a few people. I'd like to thank Lavinia Schuler for inviting me here and inviting me to attend the Brazilian Congress of Medical Genetics last week. I'd like to thank everybody in the audience for allowing me to speak in English, and I think that worked out best overall, but I hope someday maybe to be able to put something in Portuguese. Jay Lee is a technician who's worked with me for many years and actually did all the DNA sequencing. Keith Hunley is a former postdoctoral fellow. Our research is supported by the United States National Science Foundation, and also I'll take responsibility for any errors or any questions people might have. So thank you very much.
Okay, thank you very much, Jeff. Now I open for any questions you might have about his talk.
Yes.
I think you have to speak from... You have there, sorry.
Congratulations for your speech. No problem.
It's working.
Is there any relation between the high genetic variability with increased prevalence of genetic diseases? If you compare the southern countries with the northern countries?
That's a good question. So that's obviously something that ultimately has to be answered empirically. But my gut level feeling is no, that if you looked at something like diabetes, we looked at in the Native Americans, and it's very prevalent in Native Americans. Native Americans have less genetic variations, so that similarity might explain their similarity for diabetes. In the United States, diabetes is also prevalent in African Americans, but all the African Americans are genetically different from each other, but are more similar to each other with respect to that disease risk. So I think the disease risk is more likely to be explained by environmental conditions. But there are other ways that you could explain it. If there were as many, if all of those neutral mutations in Africans were matched by deleterious mutations, then there would be more disease risk too. So it's a little bit hard to say.
Just another question. It's not a nice question to do it, but I mean, I don't know about your personal opinion. Do you believe that some of our poor development has something to do with this high genetic variability? It's a kind of philosophical question, but it's I think, I don't know, Adolf Hitler would think about it, but...
Well, one of the things is if we look at genetic variation, so there are big differences in human populations, but it turns out as far as mammalian species go, humans have very little genetic variation. So if we look at, if we just compare, say, two chimpanzees with each other, they're heterozygous at many more sites than if we compare two copies of the human genome to each other. And there are only a few species, like cheetahs, I think, have less variation than people, and some endangered populations that have gone through severe bottlenecks. So it's hard to say what it means for humans, but in the broad spectrum of all mammals, genetic variation doesn't seem to be bad. It seems to be good. So there. Other questions? Yes, CJ.
Hi, Jeff. Maybe this is a naive question to ask, but all this variation you measure was in no coding genes, okay?
Right.
I wonder if you looked at the variation in the coding genes, would we find the same pattern of variation, larger variation in Africa and the smaller in the other parts of the world?
So the question is, is once again, I chose to look at non-coding regions. Would I see the same thing if I chose to look at coding regions? And once again, that's a question that will have to be answered empirically. I chose the non-coding regions because I think that they're better markers of ancestry and historical relationships and bottlenecks and those processes that operated in humans. But I would also say that any shared disease susceptibility would have to be carried on the same chromosomes that carry the non-coding variation, and so that the patterns couldn't be too different there.
Thank you.
That.
Any further questions? Okay.
This question of out of Africa or multiple evolutions everywhere, is it settled?
So there's always, that's a good question. So there's been a long-standing debate about when we look at people, did humans just evolve in Africa and then spread to the rest of the world, or did humans evolve in each region of the world sort of simultaneously and become human together? I think for the most part that is settled, and that the idea about all of the regions going in parallel can't explain the data very well. Although I do think that there are opportunities or there are instances where it looks like there was some mixing between populations as well. But I think the debate is fairly well settled. I do have one colleague, Milford Walpoff, who's a paleontologist, and he wouldn't agree with me, but I think the vast majority of people would. So.
The genetic variation was very large at the ancestral level,
yes.
200,000 years ago. Is the ancient human in Africa, is this genetic variation similar to our closest relatives, the great apes and the old world monkeys?
We still have we still have quite a bit as a species, even in our most diverse groups, like the Africans, have much less variation than the chimpanzees have. And I'm not as clear about how much variation there is in the old world monkeys. I don't think it's been studied as extensively, and certainly not for these same loci. So we have very similar data for great apes, for the ones that we've been sequencing here. So we have less variation, even in Africa, than the other primate populations.
Okay, if there is no other question, I want to thank you again, Jeffrey, for coming here and give us this very thoughtful lecture. And all of you, thanks again for being here and for your interesting questions. Okay.
Thank you.